we don't address the antibiotic resistance crisis, by 2050, 10 million people will be dying from antibiotic resistance related illnesses each year, which would be far outpacing cancer.
Thanks for coming on.
Thank you very much.
Thanks for having me.
Yeah. So Jim, when you're at home and you're going to sleep, how do you think about bacteria? Do you see it as a group of things? Like, how do you make the metaphor in your mind that is the structure of bacteria?
Yeah, it's an interesting question. I don't know that I think much about them as I'm going to sleep. But when I do attempt to imagine them, I certainly think of them as a system. I definitely don't anthropomorphize them, although I do attribute to them probably more intent than is proper.
Mm-hmm.
I certainly don't think of them as a computer system of that sort, but I don't know. I suspect it's a complicated system. I certainly have images. I mean, we look at them under the microscope all the time and have videos. And so think of them as this dynamic, it's fair, a dynamic living organism.
Now, there's so many different kinds. So, there's a lot of diversity within bacteria itself. So how do you account for that? Do you see it in a diagnostic test? Do you have because certain things could be deployed against certain other things, but you have to be able to see it. So how do you see it? And do you think they're all different?
I come from the physics world, and so I tend to look for generalities wherever I can. And probably it was best captured. I had a discussion with Marvin Minsky of AI fame a few years ago where he said that the difference in physics and biology is that physicists have 10 equations and biologists need 10,000. And I certainly even approach bacteriology, microbiology more from the physics standpoint. So I try to think of them as all being quite similar. Mm-hmm. Which is not accurate at the details, but is accurate at a higher level. Now, to get to specifics, the differences in the details are particularly important to the biologists, but they certainly become important to the point you raised, which is from a diagnostic standpoint. So from a diagnostic standpoint, we are always focusing on the differences to try to pick out what bacterium do you have or what virus do you have.
Now, in universal physics, you would have, universal truths, pretty close, except on the super subatomic level. In biology, you may not have, you may find a pathway that works with a certain strategy. So it seems that, how do you account for that? Because you have to have much more planning, or you have to outthink it in some sense, I would imagine.
Yeah, in biology, I do think there are universal truths in biology, and they have certainly percolated to the surface through the decades, through the centuries. Again, the biologists don't tend to focus on them too much, and they tend to make their passion more around finding those differences and distinctions. Again, one of the big differences on physics and biology also is the value of mathematics. There was a very famous paper by Eugene Wigner entitled "The Unreasonable Effectiveness of Mathematics in Physical Sciences," and I think I would like to soon write one on the reasonable ineffectiveness of mathematics in the life sciences. And I think it also speaks to the challenges that we have then on trying to get our hands on these organisms that do something the physical world doesn't do, which is evolve. And so the point you alluded to is that much of what we're trying to do now in the antibiotic space is to really use our wits to gain a competitive advantage over their genes, which are changing in time in response largely to what we're introducing through our wits.
Jim, for people who are just, watching and don't have the level of information and detail that you have, or maybe Jesse, could you just explain a little bit to me and for the rest of us, what it is, that work that you're doing with antibiotics and resistance, so that we can have a framework?
Yeah, so my lab's been focused on antibiotic resistance for a little over a decade now. And we view it as one of the existential crises facing humankind. The introduction of antibiotics about a century ago really was one of the major advances of humankind that helped us fight back infections that caused an awful lot of pain and death. Unfortunately, we've been overusing our antibiotics through the decades, and the number of new antibiotics being developed and approved by biotech and pharma has been dropping, particularly over the last few decades due to economic market challenges. Just about 2 years ago, we decided to team up with Regina Barzilay and Tommy Jockola, two of my faculty colleagues at MIT, who are experts in artificial intelligence, to see if we could really harness the power of AI to discover and or design new antibiotics that could be used to address this crisis. And in really short order, with an unfunded mandate, we were able to make already really very promising progress.
Jim, there's individual bacteria, there's millions of bacteria. How do you know what scale you're going to work on, and how do you measure time in that scale?
It's an interesting question. We do look at single bacteria, so we have the capability to do single cell analyses. We largely look at populations or communities of bacteria. So at an infection site, it would be largely a population of a pathogen. You'd have a single species that's problematic. Increasingly, we and others are also studying microbiomes. So these are now communities of multiple species of bacteria that are found in the environment or found in our body. So our body consists of many hundreds of different species of bacteria. Numbers have been argued about, but it appears that we're largely have likely as many bacterial cells as we have human cells. And these play an incredibly important role in both health and disease. And so we'll study both from single cell.
Which means that human beings are basically a symbiotic organism living with other organisms outside themselves. It's sort of an incredible thing to think about, yeah?
It is, and it's fascinating to see new insights that have emerged over the last decade on the important role these bugs play in things like obesity, in metabolic diseases. Maybe not surprising in things like inflammatory bowel disease, but increasingly the gut-brain axis is emerging where these bugs also appear to play a role in neurologic conditions, psychiatric conditions, as well as seem to have an interesting relationship with things such as like autism.
So let's say that you take something where we can identify that it's, say you take Crohn's disease, right? We know that that's a problem. A lot of times we don't really understand the symbiotic relationship in that some of it's doing good things. Probably the vast majority of it is doing something good. But in Crohn's disease, we know there's a certain subset of genes that are causing this problem. How do you, because you work with synthetic biology, so you don't just deploy another bug against it. You come up with a thesis and then employ ideas against it, yeah?
Yeah, so we've looked at inflammatory bowel disease, and the two dominant are Crohn's disease and ulcerative colitis. And we have considered doing a synthetic biology approach, both for therapeutic and diagnostic. Speak to the diagnostic first. We showed you could use synthetic biology to create paper-based diagnostics where you could rapidly identify what bacteria are present with somebody with IBD. Are they exhibiting an inflammatory response? And if they're in a flare-up, do they have a gut pathogen such as C. difficile that has been problematic? We in synthetic biology have also been looking about using synthetic ecosystems. So can we put together communities of bacteria that we can introduce into somebody's gut as a way to treat, for example, C. difficile, or as a way to treat ulcerative colitis? And I advise a company called Series Therapeutics that is using these synthetic ecosystems in clinical trials already with humans.
It's we see analogies about the way bacteria might work. We are told analogies about bacteria might work, but those are models of what it is, as opposed to being the real thing. How do you, it seems like it's so incredibly complex, and you have to have a thesis to attack a problem. How do you think about that at the outset?
Yeah, it really depends on the problem. The way we've approached it over the last 20 years, broadly looking at bacteria and antibiotics over the last 10 years, has really been from an engineering perspective. And so we've looked to see, can we engineer wet circuits similar to the way an electrical engineer would engineer electrical circuits, introducing these into bacteria, uploading them, encoding them in DNA, uploading them, and seeing if we can now program the bacteria as a form of living technology, either then to serve as a diagnostic or a therapeutic. We've also then looked to these bacteria as communities, that they can be viewed as multiple individuals that are interacting, communicating with each other, helping each other out, supporting each other, and sometimes sacrificing themselves to help the larger community.
What tools do you use to do that? Is that CRISPR? Is that, what tools are required to do that?
The tools of synthetic biology bring modeling tools and design tools from engineering. And then in the early days, we used the enzymatic tools from genetic engineering that enable us to cut and paste bits of DNA together into circuits that can then be introduced with electrical signals or other chemistry into bacterial cells. More recently, we've been using CRISPR, which is a new set of enzymes that enable us in a very directed manner to modify DNA of living organisms, as well as to detect, for example, RNA or DNA outside of organisms. So we have developed, among other things, CRISPR-based diagnostics to detect pathogens, including COVID-19, SARS-CoV-2.
We, over the last 10 years, how far has synthetic biology come? George Church's work, how far have we pushed, since then? Are we flowering as a group, and are we going to see great advances from that?
So synthetic biology is about 20 years old. And I think we're at a very interesting transition point for the field, where the field is moving from being an emerging field to a maturing field. Still relatively small from an academic standpoint, a few hundred labs. Most of the activity appears to be in the commercial space. So now there are many more companies than there are academic labs. And I think we're going to see an explosion in this next decade of synthetic biology advances in biomedicine, in the environment and sustainability, back toward bioenergy, but also in the consumer space.
When we see things like immunotherapy exploding and we see proton therapy exploding, is there a role for synthetic biology in conjunction with those things?
So in conjunction with immunotherapy from a cell therapy and gene therapy standpoint, absolutely. I think where synthetic biology is coming into play is really giving us unprecedented control over the expression of the genes that might be part of gene therapy or cell therapy. So in gene therapy, it's largely been a focus the last 2 decades to try to get very safe and effective delivery of DNA constructs into somebody's cells so that you could turn on a gene or express a gene that might be missing or defective. Synthetic biology is now giving us the ability that you could turn that on only during certain portions of the day, or you can tune it depending upon the level of the person's condition, or you can turn it on only in certain cells or certain tissue types, giving us now much greater control to really begin to address disease in fairly sophisticated ways.
Can I just push you a little bit for that? For people who don't really understand, there's a fear of synthetic biology, and now you're talking about genes. This idea that you could turn a gene, or you could have more control, I guess. And what would be an application for that? Would that be something in diabetes or some other conditions where we can start to help, monitor people's health? Because I think what we want to do is have you kind of talk around what is possible, right, for health and for well-being for people, and what's the application process in a person for this work?
Yeah, I think the promise of synthetic biology is broadly this idea of control. And control is this notion that you can now program genetic constructs or living cells so they can sense something in their environment, make a decision, and act on that environment. And in the context of health, the idea is, could we now create adaptive medicines that are living in nature so that you could have an engineered cell that's introduced that can seek out the infection site or the disease site, make a measurement, and produce the therapeutic that's needed at that site, and only produce it at that site, and produce it at the right dose. So one could envision in diabetes, can you create cells that could replace a misfunctioning or missing pancreas or pancreatic cells that could sense the level of blood glucose and appropriately produce insulin as needed? And this is part of the promise of synthetic biology.
And what about we talked a lot about bacterial resistance, And so how can some of the techniques that you're working on, I know for myself, I've taken way too many antibiotics, and we hear that, how can this work that you're doing work to combat that?
And I think two ways. One is we are using synthetic biology to come up with means to overcome resistance and to resensitize bacterial cells that have become resistant, making them susceptible to an existing antibiotic. And with Tim Liu, how we did this is we actually engineered bacteriophage. So bacteriophage are viruses that only go after bacteria. They were used as antibacterials in the former Soviet Union. They come back in vogue, and we showed that we could engineer them using synthetic biology so that they would produce proteins that would make a resistant bacteria now able to be killed by the antibiotic to which it had been resistant. And it was able, really remarkable, to overcome that resistance. The second means goes back to the artificial intelligence. And there the idea is, can we come up with completely new antibiotics that the bacteria have never seen and for which no resistance had yet arisen, so that we can stay ahead of them and use the tools of AI to come up with these completely new molecules that now we could prescribe. And in time, resistance will grow. But the notion is that that platform could be used to continually innovate and produce more and more antibiotics that we just roll out as needed in the face of new resistance.
How do you think about dosage? Because, a lot of the problem of bacteria, the bacterial resistance comes from just overuse of antibiotics, but also not necessarily understanding the dosage that would have an effect. Do we have to does this, bacterial resistance call upon us to rethink how much of a drug and how we think about it?
It does. And a discovery we made in our lab, now it's almost a decade ago, was intriguing in that we showed that low levels of antibiotics, those that did not kill the bacteria, were highly problematic because it made them stronger. And it did so, and that the antibiotics themselves became mutagens. So if the antibiotic didn't kill the bacteria, it increased its mutation rate and thus actually further promoted resistance. So for example, if you're using antibacterial soaps at home or wipes, you actually may be promoting resistance in your microenvironment. And so it becomes highly problematic. So we need to think differently on dose. But there's the other end of antibiotics, which is underappreciated, which is many antibiotics exhibit toxicity. So you check in Wikipedia, you talk to med students, and the general notion is that antibiotics don't kill human cells, they kill bacterial cells. That's true at almost all clinically used doses and supported by screening. What's not appreciated is that antibiotics do impact human cells at clinical doses. They don't kill them, so we screen them so that we make sure they don't kill off the human cell. But they do impact them, and we uncovered that they, for example, interact with our mitochondria, which are organelles in our cells that are thought to be of bacterial origin and that are key for producing energy. And we showed that antibiotics at clinical levels will interact with the mitochondria, producing oxidative stress, which can lead to mutations in our cells. So there's this juggle between making sure you don't have too high a dose or too toxic level of antibiotic that could significantly impact and adversely affect your cells.
You alluded to this earlier, but the financial models, right? Is there enough money available for diagnostic tools? Synthetic biology has an incredible capacity for doing diagnostic tools, but diagnostic tools are not a place where there's a lot of, they don't cost much, and, reimbursement is a factor. So are we holding ourselves back from our best selves because we don't have the financial model of this part of it, figured out yet?
Yeah, I think that both diagnostics and antibiotics are problematic from a development standpoint. Diagnostics don't fit the model for the venture capitalists, in that a diagnostics company, the value really comes from just a multiple of the revenue of the products that you have out approved. And so you have a very fixed and easy model. Whereas a therapeutics company, you can sell hope for 10 to 20 years without ever selling a product. And so you can get 10, 20, 50, 100 multiple over your initial investment. And that's much more attractive to many venture capitalists. The pandemic, the current pandemic, has dramatically changed that, in that now people recognize that We've not put in sufficient resources into our diagnostics, and we need more innovation and we need a better model. The US government did not really get behind that at the level they needed to, in my view. They did from the vaccine, so Operation Warp Speed was marvelous. They didn't have an equivalent for the diagnostics. In the antibiotics space, the market is horribly broken in that the amount of money it takes to develop an antibiotic is on par with what it takes to produce or develop a blood pressure drug or a cancer drug. An antibiotic is usually sold for a very cheap cost. You use it only over 5 days, maybe 7 days. Blood pressure drug, you could sell much higher, you take it for the rest of your life. Cancer drug, you could sell even higher. Hopefully you don't have to take it for the rest of your life, but you really would take it only 5 days. It's going to be over the course of many weeks, months, if not years. And so I have many colleagues in the pharmaceutical company. I don't fault them in that their business model is set up that it makes better sense to go after blood pressure drugs and cancer drugs. But there's a dramatic need on the antibiotic resistance side that we are not addressing. I spoke of the existential crisis. There's a group in the UK that estimates if we don't address the antibiotic resistance crisis, by 2050, 10 million people will be dying from antibiotic resistance related illnesses each year, which would be far outpacing cancer.
You know, our COVID resistance, our ability to come together around the pandemic, in some senses was good, you know, in terms we have this drug, but cooperation around the world has been very, very spotty and difficult. You know, a crisis like this, you know, does it lead us to a step forward in how to solve some of these existential threats? I mean, synthetic biology by itself is an existential threat. You know, it's like nuclear proliferation's an existential threat. You know, you go on and on. Climate itself is an existential threat. So does something like this give us the way to start to think about some of those threats and how to take them down?
I think it does, and I think it does in two ways. One is I do think that the pandemic, which has been horrible, has some positive outcomes, one of which I think it shows that these larger scale crises do have dramatic impacts on the economy, in addition to just general well-being. And it sets back the economies. And I think nation states have woken up that we need to address public goods more collectively and with greater effort. But I also think that private industry and philanthropists have recognized that there's also a need to address these large scales, whether it's the next pandemic, which is coming. We don't know when, we don't know from where, but it's coming. But let alone climate change, which similarly in a very short period of time will set us back on our ability to function well as a society. The second is that I do think what unfortunately could be emerging as a quasi-lost generation of our youth who are impact a critical point in their development by the current pandemic, I think are nonetheless being galvanized to consider going into infectious disease, as well as these other larger issues like climate change, to see where there really is a need and impact, and kind of sitting back to see that doesn't matter how much wealth you have. It really matters having meaning in your life, and can you make a difference? And I, for one, am really encouraged to see a number of young people seeing infectious disease as a tractable area to come in to really be innovative and attempt to be impactful.
You know, in the last 20 years, we've seen a great commitment to shareholder value. Everything is shareholder value. But doesn't this sort of beg the question about whether some of the, you know, sort of resource dollars should be going into something like bacterial resistance because it is an existential threat, and maybe that doesn't return as high a rate of return, but is just as important in terms of the human species?
I think so. Yeah, I mean, I'm beginning to see a shift from shareholders to stakeholders. There still is the challenge on what are the models, and can you get the right motivated talent? So if you're trying to bring in that 30-year-old innovative biotech person, are they going to join your nonprofit or your for-profit, but with only marginal gains, as a key scientist versus this CRISPR company that, you know, could return 100-fold on their options? And it's a matter of how to find that right balance and motivate folks. I take some heart in, I think back decades ago, of the Peace Corps and a greater notion of public service and national service. And I do think people want to have meaning and impact. And again, I'm hoping that what we're all going through will help motivate folks to take their eye off profit motive and move more toward impact and impact investing and commitment of their time.
You know, Jim, when you talk about, I'm sorry, Jesse, but when you talk about the sort of existential threat of bacterial resistant drugs and what is coming upon us potentially. Are there a finite amount of bacteria? Like, is everything already there, or can new forms of, bacteria be created in our world, or as a result of terrible climate conditions? And again, I know I'm not a scientist. I don't know anything about this, but when you're saying like there's potentially some really bad times ahead, where are all of these bacteria coming from?
It's an interesting question. So some are coming from our microbiomes, some are coming from the animal world, some are coming from the environment. Resistance, I think, will only generally keep going up, meaning increasing. And the bacteria want to replicate. Again, I'm now anthropomizing them, but they would like to replicate. They like to pass on their genes. And so they will mutate, and those that have an advantage will continue to propagate. And unfortunately, it gets compounded in that bacteria will also share plasmids, so rings of DNA, and exchange these between species, across. And these rings of DNA can include antibiotic resistance genes, and they can begin to build up. And so you get these super pathogens are often ones where they've got these rings of DNA. And so this problem is a problem that's going to continue to grow. And unfortunately, it's being exacerbated by our overuse of antibiotics. If many of your listeners who go to see their doctor, maybe they have a sinus infection or a cold, they demand an antibiotic, which largely probably not have an effect. Farmers are using antibiotics as prophylactics for both protection against potential disease, but also as a growth incentive for their livestock. And we just need to find alternatives in each of these so that we can reduce the pressure for increased resistance.
Can that be inherited? Is it
The resistance?
Can it be inherited to a baby?
So the baby bacteria, absolutely. So when the bacteria divides, it's going to pass on the resistance to the next. Amongst humans, if there are resistant bugs that are in your microbiome, then I think it is likely that it can be transferred from mother to baby during the birth process, depending upon where the bacterium is.
But it's not in the genetic code.
It wouldn't be in our genetic code, no. But it would be in our microbiomes.
Because you've built a lot of companies, what takes an idea from research to I want to build a company?
I think in many cases it's seeing that the research idea or the output really has a direct application, and that it fills a need that has not been addressed. In our case, we're largely looking at biomedicine. So it would be seeing, for example, that we can use CRISPR and synthetic biology to create rapid diagnostics. We decide, okay, we think this could really form the basis because we can rapidly develop rapid diagnostics that could be useful in outbreaks, epidemics, and pandemics, but could also be useful for treating general conditions. So it's a matter of cycling through, do we think there's potential here? And then, as with so many things, the most critical is actually finding the right team that can act out on the vision, strategize appropriately. The technology, the science, in my experience, has only been a very small part of the story, maybe 5 to 10%. And it's really the team, the business team, the scientific team that makes all the difference.
When you see things like immunotherapy exploding in terms of all the different applications, there's so many applications now you can't even keep up with them. Do you see a similar thing happening here?
I do. I think we're at a fascinating point where 20 years into the field, the last few years, we've seen a real shift towards using synthetic biology to create new classes of medicines around this idea of living therapeutics. And that includes treatment of rare genetic metabolic disorders, tumors, inflammatory bowel disease by a company called Synlogic that we helped launch, to doing next generation cell therapy and gene therapies by another company called Senti Biosciences, to those synthetic ecosystems such as Series cells. So I think we're opening up whole new possibilities, which is so exciting. And the level of control allows us both to increase efficacy while also trying to keep things safe, if not even improving their safety profile.
The artificial intelligence, when you apply that, how do you apply it? Is there a data set that or do you take the diagnostic test and put it into, a computer and that's the AI? Or what's the underlying data set to help look for an idea? And what do you train it with? Or do you train it at all?
Yeah, maybe I'll give our example with our antibiotics effort that we reported on just a few months ago to give you a sense. What we did was very straightforward. We actually put together a training set of molecules to start. So we brought together 2,500 molecules, drugs. This included 1,700 FDA-approved drugs that included the known universe of antibiotics, about 300, plus 800 natural compounds, so molecules you'd find in nature that might be made by plants or insects. Took those 2,500 molecules and applied them to E. coli. And E. coli is a gut pathogen that's really the commonly used in labs. It causes urinary tract infections, so it's a pathogen as well. Applied each of those to E. coli and asked which of those molecules exhibited antibacterial activity, meaning which of the ones inhibited the growth of E. coli. We took that data as training data, and we took the information on each molecule, which was its structure. So the type, when you think of your chemistry classes, the chemical structure, and we fed that information to a computer model known as a deep neural network. And we trained this deep neural network that's based weakly on the way the brain is structured to associate atom by atom and molecule by molecule of each compound that we looked at with whether or not it exhibited antibacterial activity or not. So by training it through many rounds, we arrived at a computer model that we could then apply to new sets of molecules to see which ones are predicted based on the training to make for good antibiotics.
How long did that take?
The collection of the data took on order a few weeks, but the training took only a matter of a couple of days.
Mm-hmm.
But more stunningly, we then were able to take the trained model and apply it to very large libraries of compounds that would be otherwise impossible to explore. So for example, we applied it to a library of 1.5 billion molecules that were present in a computer that you could never screen. Empirically, you just would never have the time. We were able to look at 10% of that, so 107 million molecules in just 3 days, screening for new antibiotics.
And how many candidates did you get?
We identified hundreds of promising candidates, and we went the next step. We then asked two additional criteria of the model. One was to predict only those molecules that don't look like existing antibiotics.
Right.
So for the last couple of decades, people just keep rediscovering what we already have. And second, we want to identify molecules that look to be not toxic to humans.
Right.
And we got that down to several hundred. We were able to purchase or synthesize quickly a couple dozen, which we tested, and 8 of which turned out to be completely new antibiotics.
How long does that take? I mean, the kind of to go from that testing to actually getting into the hands of people or getting into places where we can use it. The time.
So the discovery efforts that we had are really, it can be done in a matter of a small number of weeks. So really in a small number of months, if we faced a new pathogen, we could identify that molecule. Now the challenge is to do the clinical trials.
Now, do you get cooperation by the drug companies to look at those molecules? Do you need it to look at all those billion different things?
We don't need cooperation from them, no. So in fact, that was a publicly available data set. And then we would need to purchase them from other vendors or synthesize them ourselves. But that set was publicly available. The pharmaceutical companies have other data sets, and we're looking to see if we can partner with them to look at some of theirs.
You started about clinical trials, though, and we know that doesn't that take a long time? So how can we speed up that process?
Yeah, so usually you have to do your 3 phases. Phase 1, which is largely looking at safety, make sure you don't do any harm. Then you'll do an initial phase 2 around efficacy, does your drug work? And then you do a larger randomized phase 3. 2 things of note. One is what we saw with Operation Warp Speed is that under certain conditions, we can dramatically speed up the trials. And so for the vaccines, we saw multiple 30,000 patient trials that were done in record time. I don't think we're at the need yet on the antibiotics to do that, but what we're doing is we are also looking to see, can we identify rare conditions or orphan conditions of an untreatable resistant infection that would allow us to set up a much shorter trial that could be done with smaller number of patients over a brief period of time for much less money that would dramatically reduce the cost, that we could then challenge our AI model to come up with molecules that could treat those infections.
Culturally and intellectually, that sounds like, of course you should be doing that. You have the, the potential to make people better. What has to happen in our environment, socially, to get people to say, let's move more quickly? Like you said, this most recent pandemic, we're seeing that, but what else needs to change?
I mean, in your mind, is this pandemic going to be like when HIV occurred and people went and protested at Congress, and we got some rapid development of drugs based on that? Is this a opportunity for the future to speed up biomedical research?
I do think it represents an opportunity. So a couple points to that. I do think what we saw with HIV was marvelous on the response, both of certain leaders in government. I think the Hollywood community, entertainment community, sports community really rallied, got public interest, and applied pressure. And it did two things of note. It led to dramatic resources being levied for research and development against the disease, and encouraged young minds to go into the space. I think we need to see that again on larger infectious disease notions. I don't see that happening yet around antibiotic resistance. So, for example, there is no major foundation dedicated to it. There is no month, there is no day, there is no march, there is no major celebrity backing it. Yet we've all lost family members to bacterial infections. And yet it seemed, often it's your relative goes into a hospital for something else, but then catches a bug and ends up dying. But you think of more of why they went into the hospital versus what happened. And so I think we need more public awareness, public engagement in order to get people rallied around it. We need to change some of the needs and the size requirements of the trials. We need more research to come in. We need to change the economic model. Again, it's, if you want to make money, it's better to put your money into one of the internet firms or online firms than it is an antibiotics company. But if you want to have an impact, to support these young companies or efforts is where it's really needed and can make a difference. And I think, Jesse, you made the allusion earlier in our discussion, you can still make a return. And minimum, even from a philanthropic standpoint, it could be a huge benefit for relatively modest investment. So the time is now to begin to address what is going to just keep growing as a problem.
Do you, I recently did a project for MD Anderson where we looked back over the 75 years of medical innovation for cancer research, and it's really astounding. A lot of times we don't think of it that way. We think that not enough is happening. But are you optimistic for the future that we will iron these things out and in our time here push them along?
Yes, I am. I mean, I tend to be an optimist, but I very much believe in innovative spirit, and I do think the need is there. I do think certain groups are ringing the bell. The United Kingdom, for one, and some groups in the U.S. People aren't responding yet, but I think we have the innovative capacity to address this, and we just need to rally more folks at a scale to make that difference. Again, early discussion, I do think the discovery of antibiotics remains one of the major contributions of humankind to humankind. And you think back, it was 100 years ago, and fascinatingly, the golden age of antibiotic discovery was in the 40s, 50s, and 60s, before the molecular biology revolution, before the biotech revolution, before the genomic revolution, before the AI revolution. And so we're poised to, I think, discover, explore, and design a chemical space in ways that our predecessors just didn't have the capacity to. But we need to get the right resources, both capital and intellectual, into this space.
Well, Jim, it's been an incredible conversation. We deeply appreciate you coming on and sharing with us.
Well, thanks for having me. I really enjoyed talking with both of you.
Thank you so much. Thank you so much.
Thank you, Jim.